inbreeding depression, and so it kind of rhymes, so I think that's why I'm in. So thanks for coming. So this is the work of a student. Okay, so inbreeding depression, as I'm sure everybody knows, is the reduction in fitness of inbreds relative to outbreds, and you can think of it as the strength of selection acting against inbred types. Inbreeding depression is a key factor in mating system evolution. It's important for assessing extinction risk, and it's probably the simplest and easiest way to provide some evidence of segregating genetic variation for fitness in populations. And so for all these three reasons, evolutionary biologists, Darwin, have studied inbreeding depression, and there's hundreds, if not thousands, of estimates of inbreeding depression across a wide variety of taxa. And so we know a few things from all those studies. First, we know that inbreeding depression is really common. You almost always find it when you look for it. But we also know that it's highly variable in its strength. And we see that variation among species or among populations of the same species. But we also see a different kind of variation. We see variation from a single population when we measure it in different environments. And so what I want to think about today, talk about is why is inbreeding depression variable? So here's start with a very simple population genetics model and calculate inbreeding depression. And if you do that, you find this simple expression that tells you that inbreeding depression depends on the degree of inbreeding, which is typically known when you do an inbreeding depression study, but it also depends on the recessivity of the deleterious allele, the strength of selection S, and the level of polymorphism. And so we can use this theoretical framework to think about why there's variation in inbreeding depression. So for example, when we compare different populations, if they differ in the level of polymorphism, then we might expect differences in the levels of inbreeding depression. And people have said this before. For example, populations that have gone through bottlenecks will have reduced levels of polymorphism, and thus we expect reduced levels of inbreeding depression, and there have been some really nice studies showing that. But that's not what I want to talk about today. Instead, I want to talk about when you take the same population and measure it in different environments, you're sort of holding the level of polymorphism constant, and what might differ are something about H and S. So that's a one locus model. If you make a multi-locus model and some simplifying assumptions, that simple equation becomes a bit more complicated, but at least the part in black is basically the same as the one locus version. And my main point of showing you that is just to show you S bar that inbreeding depression will be stronger in where mean S is greater. So in some sense, we can turn the question of in what environments will inbreeding depression be stronger to asking this question: in what type of environments do we expect mean selection to be stronger? Now, I do want to point out there's all these other parts to this equation. So we don't expect the relationship between S bar and inbreeding depression to be a perfect relationship. It's going to be somewhat blurry. But on average, we do expect that in environments where mean S is stronger for inbreeding depression to be greater. Okay, so in what environments is S bar greater? Well, one intuitively appealing idea is that stress increases selection on average across mutations. In my opinion, there's sort of two problems with that intuitively appealing idea. First, I'm aware of no theory that predicts S bar. A lot of people don't care about theory, they care about data, but that's where the second problem comes in. So probably the most direct way of addressing this question is to go and look at individual sets of individual mutations and measure them in multiple environments. And that's been done now in a number of studies. And a few years ago, Mike Whitlock and I reviewed that data, and we found the results are really mixed. That is, in some cases, you find the average selection across a set of mutations is stronger in a stressful environment, but in other cases, you find the opposite, and in other cases, you find no change in mean selection. Individual mutations become more strongly or weakly selected, but there's no change in the mean, or very little change. But in some environments, you do see mean S being stronger, and one one thing that emerged from reviewing that was the idea that that mean S was stronger in environments where fitness was more strongly density dependent. And here I'm using density dependence to simply mean the sensitivity of absolute fitness to density. And so the first question is, well, why should that be true? And I think that there's a very simple reason for that, which is that unlike some other kinds of stress, the effects of density don't fall equally on everyone. That is, the best individuals kind of get out ahead of the crowd. They start consuming resources. They start producing waste products in the environment. And the best individuals that start off with a little bit of an advantage kind of make the environment worse for those around them. This is exactly the same logic that's behind the idea of dominance and suppression in the plant competition literature. So based on that idea, we have this hypothesis that if increased density dependence increases mean selection, then increased density dependence should also increase inbreeding depression. And so that's the idea that Lee wanted to test, and essentially the goal is pretty simple, is that you can measure inbreeding depression across a range of environments that vary in how stressful they are and how density dependent they are. Okay, so this is what Lee did. Using the software melanogaster, he created 90 lines that were made to be homozygous for chromosome 3, which represents 40% of the genome. So this is a cartoon version of the 90 lines showing just chromosome 3. And you can do crosses within lines to yield inbred eggs, and you can do crosses between lines to create a bunch of outbred eggs. And then we can use those sets of eggs to do 4 different types of assays for each environment. So he did assays that contained 130 outbred eggs along with 130 marked competitors, or did the same thing with inbred eggs and competitors. And then in addition to those 2 treatments, he, using outbred eggs, did it at a slightly lower density and a slightly higher density. And then did 30 replicates per assay. And then did this in 22 different environments that range in their chemical composition, thermal regime, nutritional quality of the food, the viscosity of the food, and the amount of water availability. And data I'll show you represents the counting many flies. And I want to point out, so he's done this in 22 environments. There have been a bunch of studies that measure inbreeding depression in multiple environments. Almost all of those measure them in 2 or possibly 3 environments, and I'm not aware of any that do it in more than about 5 or 6. So this is quite a broad set of environments. And here's a cartoon of the kind of data that he has. So he's measuring survival egg to adult survivorship as our measure of fitness, and done it at these 3 densities. And we define stress as it's been simply defined in this type of literature as the reduction in fitness, in our case survival, below the theoretical maximum, so at our focal density. So that's the measure of stressfulness. Density dependence is as I said, the sensitivity of fitness to density, so it's just the slope of that line. And so those might be the data for one environment, the green, let's call it the green environment. Here's data, cartoon data for a purple environment, and we would say this has a similar level of stress, but the second environment is more strongly density dependent. Going back to the green environment, remember for the focal density, we've measured outbred flies and also inbred flies, and the difference between those 2 tells us something about inbreeding depression. Okay, so what we want to do is characterize these 22 environments with respect to how stressful they are and how density dependent they are, and then look at the relationship with inbreeding depression. So first thing, for this set of environments, how much variation do we have in stressfulness and in density dependence? Well, we have quite a lot of variation in how stressful the environments are, about a 6-fold range of stressfulness. And I've just put the environments sort of closest to our standard lab conditions in purple. It's kind of a reference point. We also have about a 6 or 7-fold range in variation for how density dependent the environments are. But at least amongst this set of environments, stressfulness and density dependence are not particularly well correlated. There's a non-significant correlation between the two in this data set. So now thinking about the relationship with inbreeding depression. So is inbreeding depression related to stress? That's a question people have asked about before. And in this data set, the answer is no, not really. There's a non-significant positive correlation between them. And I should point out that all the statistics here have been done in two ways. First, the sort of straightforward way of using Pearson moment correlation, but there's some problems with that. First of all, the distributional assumptions aren't great, and perhaps worse than that, there's some correlated measurement errors in the data structure. So you can build a big likelihood model that accounts for all that, and it tells you almost exactly the same thing in all the cases. In fact, you waste a lot of time doing it. Anyways, so inbreeding depression and stressfulness are not particularly correlated. What about with density dependence? And here we do find that there is a stronger and significant correlation between inbreeding depression and the degree of density dependence. And of course, you can do the full model where you include both stress and density dependence, and what you find is there's no main effect of stress. And in fact, the earlier correlation that you saw is probably the extent to which there wasn't any correlation with stress and density dependence when I just looked at those two together, or between stress and inbreeding depression. That relationship there was probably mostly driven by the relationship between stress and density dependence. So when you put them both in the model, the point estimate of the effect of stress is almost zero. There's a much stronger effect of density dependence on stress. There does also appear to be this interaction effect that seems to be driven by these 2 points, but if you look at the data, there's mostly this fairly strong trend towards increasing inbreeding depression with density dependence. And so just to conclude, we found, as others have found, that inbreeding depression varies a lot depending on the environment you measured in. So in our data sets, about a 4-fold range of variation. And most importantly, from my perspective, is that inbreeding depression seems to be correlated to density dependence. And that's consistent with this idea